All right. So we left off on Tuesday talking about the different types of infection, in terms of being acute or chronic. Latent, remember, latent just means that the virus, typically latent diseases are virus related. But ultimately, they're going to be when the virus doesn't actually leave the body, it just goes into hiding, and it can be there for an indeterminate amount of time. Some of the other bacteria-related would be things like tuberculosis and leprosy that can actually go into latency too. So, new information, we got the distribution of a pathogen. This is important for being able to understand what level of triage essentially the patient's in. If it's going to be a localized infection, that's something that somebody comes in, they have some sort of wound, typically, and the wound is infected, that's going to be localized. And that's typically going to be something that's easily handled for the most part. It's going to be, microbes are going to be limited to that small area. When it becomes problematic is when it goes systemic. And so systemic just means that it's disseminated throughout the entire body. And those are much more serious. And so the different suffixes that we use for different types of infections are going to be emia. So if it ends in emia, it just means it's gotten into the blood. And there's different variations of it. If it's bacteria related, it's bacteremia. If it's a toxin, as we'll talk about a lot of different types of toxins, toxins are going to be toxemia, viruses, viremia, and then septicemia is typically the one we use. We usually don't differentiate between all of them. Sepsis, septic or septicemia is going to be the term that you'll hear the most often. So how do we actually know what infectious agent is causing what disease? How do we know that influenza actually causes the flu? How do we know that the Staphylococcus aureus causes many different types of diseases, but ultimately sepsis is one of the bigger ones? Well, we have to use what's called Koch's postulates, and this goes back to chapter 1 in Robert Koch, and the way he was able to determine using a very systemic science approach to understanding that Bacillus anthracis was the cause of anthrax. And again, this was done in the late 1800s. And so the process was novel at the time, and we still use it to this day. It's still a process that's used every time we have a new virus, new bacteria, new fungus. We go through Koch's postulates. And so the idea is that the microbe has to be present in every single case of the disease. So if somebody comes in with suspected anthrax, we have to be able to pull Bacillus anthracis out of the system in order to verify that it's truly anthrax. So when we're looking at it from a research angle, we're talking about things like mice, animal models, maybe primates, the other different types of models we use. It doesn't always have to be a mouse. It's going to be whatever model is important for that particular disease. And so the microbe has to be present. The organism must be grown in pure culture. So once we pull the organism out of the animal, we have to be able to grow it in a petri plate, typically, or some sort of cell culture. And then it has to be able to isolate it from a pure culture. So bacteria, sometimes when we pull it out of, like, say, a nasal swab or something like this, it's going to be a mixture of different bacteria. So it's a little bit of a process for microbiologists or a med tech person to go through and tease it all apart. Once we have a pure culture where we know it's just that particular microbe, then we're going to introduce it back into an animal model. And the anticipation then is that we should get the same exact disease when we put that microbe back in. So we extract it out of the animal model, we grow it up in a petri plate, we put it back into the animal model, and then we pull it out one more time. If we get that process and we're able to confirm it, then we can say that with pretty strong conviction that particular microbe is what is actually causing that particular disease. And so to this day, we still have to follow these postulates in order to verify particular bacteria and viruses. Now, limitations are going to be that some organisms it's just impossible to grow in a laboratory setting, still to this day. For instance, Mycobacterium leprae, the leprosy-causing bacterium. Very difficult to grow in the lab. We actually have to use armadillos, and they really only grows in the foot pads of armadillos. It's a very strange bacteria, so that's a very problematic system to have in a laboratory. Things like syphilis. Syphilis grows inside cells. It's a bacteria. We'll cover it when we get to the genitourinary tract infections. It's a bacteria, but it's an obligate intracellular bacteria, so it actually grows inside cells. So again, trying to mimic that in a laboratory setting in vitro. Remember, in vitro means outside the body. It actually truly translates into in glass, and versus in vivo, which is in life. And so when we're actually trying to grow some of these different microbes in vitro, it becomes really difficult, if not impossible, to get the situation just right. And so sometimes infected individuals are not always going to have symptoms. So things like polio, you can actually have a very mild subclinical case of polio, and so therefore it would be difficult to actually show that that person was infected. Some are actually going to be polymicrobial, so that means it's more than one that actually causes the. Everybody sign
Multiple species, right?
Huh?
Multiple species.
Yeah, multiple species are actually causing it. So we'll see this when we're doing. We'll see this when we're doing the digestive diseases. One of the very first ones we're going to cover is cavities. You know, you don't think about cavities being an infectious disease, but it truly is because it's actually caused by bacteria. It's multiple bacteria that come together to cause these acute necrotizing gingivitis or cavities. You know, so this actually say it's just one microbe is not true. And so again, that would fall away from Koch's postulates. And then sometimes there's just not a suitable animal host. Again, the idea that up until we started using ferrets, there really wasn't a good model for influenza. Flu, to actually get the same symptoms as a human has, it was really difficult to find. And even things like salmonella. We were just talking about the meat recall that just happened today. It's salmonella. Well, you put salmonella into a human, it's basically a self-limiting, in somebody that's healthy, it's a self-limiting disease where it's going to cure itself within a couple of days. Your immune system's going to get in there and take care of it. I give salmonella to a mouse, it goes systemic and kills it. That's not a good model, right? It's not the same exact model as what we see in humans. So again, some animal models, some hosts really just don't exist to be able to fulfill the postulates. Now, some of the things that we do today that weren't available back in the 1800s when this was first put together is something called molecular Koch's postulates, which is being able to actually take genes out. So remember, Tuesday when we were talking about virulence factors, and these are the parts of the bacteria, virus, fungus that are responsible for actually causing the disease. So we'll see some of them as we move forward here. But things like a toxin, the actual processing of making that toxin, that would be a virulence factor. Well, if I could go into that microbe and actually remove that gene, figure out what gene is involved with the toxin, remove it, then we'd actually be following molecular Koch's postulates, where we take the virulence factor gene or whatever product that we're actually trying to remove, we take it away, we see if we get the same disease outcome when we actually have this genetically modified microbe. We'll see if we get the same outcome, and if whatever we end up getting, hopefully, we'll actually see a decrease in virulence, and then we have to put that gene right back in. And if we get that same increase back to normal, then we know that gene's important. And so that's actually things that we do more or less today to try to understand, okay, yes, we know salmonella causes food poisoning, but what is it about salmonella that's actually important? What genes actually allow it to cause disease? And if we remove them, do they suddenly become benign? And so that's what we're talking about there. Molecular Koch's postulates is some of the new techniques that we have available to us today that weren't available 100 plus years ago. So here we're talking about mechanisms. This is going to be very generic. The next few slides are going to be very generic. A couple examples. I want you to really just focus on not the examples per se, but actually the types of characteristics of pathogenesis that we see, that basically I can take all the different microbes and I can clump them together, underneath a lot of these different characteristics. And the reason why I say they're generic is that when we get up into the later chapters, we're going to see a lot of these microbes individually, and then we're going to talk about what it is that makes them pathogenic. Here, we're talking about all the major categories of things that we see that are pathogenic. Yes?
Pathogenesis, what's the definition?
Pathogenesis is actually the process of causing disease, how that pathogen causes disease. So, produce toxins that are ingested. So in this case, something like Clostridium botulinum. We now use it, in a very controlled manner. We use it as Botox, but the actual bacterium itself produces a toxin. And when we first started having problems with it was in the 1950s and 60s and into the 70s. When we first started canning food. To us, nowadays, we take canning as just, we go to the grocery and buy cans of food. Well, that's relatively novel. At least for our parents' generation, maybe your grandparents' generation, that's relatively novel as a concept. And when we first started canning, we didn't quite understand the dangers that were involved with that. And so Clostridium botulinum is actually a bacterium that likes to live in anaerobic conditions. Well, there's nothing more anaerobic without oxygen, right, than being in a sealed can. Bacteria loves it. We grow up in really high levels, and if you ever heard the idea of not buying a dented can, that comes from Clostridium botulinum. It was the idea that if the can was dented, that assumed that there was actually air pressure building up underneath it because the microbe produces byproducts and causes the can to start denting and contorting. And it was very scary because before we started realizing that, people that would then eat that food, they'd actually eat the toxin, and the toxin would be very deadly. It could potentially kill you. And so, here nowadays, we actually have, as we've seen in Chapter 5, we have a lot of regulations against that. We actually have the retort systems that sterilize food to a pretty solid level to minimize any sort of dangers involved with Clostridium botulinum now in canned food. But it is still something to just keep in the back of your mind. Colonizing mucous membranes. So here it actually gets inside your body and then produces a toxin. And so these would be things like Corneobacterium diphtheria, causes diphtheria. Also, E. coli O157. This is the one that's the most dangerous of all the E. colis. There's literally tens of thousands of different species of E. coli. Most of them are benign, but this O157H7 is actually one that caused a major outbreak at a Jack in the Box in Oregon, I believe, many years ago, and killed quite a few people. Vibrio cholera, we'll get into cholera as a disease, but again, all three of those produce a toxin. It's not necessarily the bacteria that's making you sick, but the toxin once it gets inside. So notice the difference between 1 and 2 there. You can actually ingest the toxin of botulinum without actually ingesting the bacteria. It would produce sickness. The next one down, it has to get inside you, establish residency in the mucous membranes, and then produce that toxin and cause sickness. Invading the host tissues and avoiding the defenses. We've seen a lot of that in Chapter 14 and 15, where some of these, as I was telling you before, it's like a strategy game where sometimes the microbe actually invites itself to be phagocytized, and then it can produce sickness afterwards. It has ways to avoid it. And so that would be things like tuberculosis, pestis, which causes the plague, and Salmonella enterica, which is one of the other Salmonella strains. Invading the host tissue and then producing toxins. So here, again, just another example. Notice how toxins keep showing up here. Toxins are a big deal for a lot of these different infectious agents. All right, so establishing an infection. Very first step, as I pointed out before, they have to adhere. Say you ingest some food that's got some, infected with some sort of pathogen. Well, if it gets down your stomach, gets chewed up by the stomach acids, the lysozymes in your saliva, goes through your digestive system, never attaches, whatever may be left, and just goes out the other side, you're never going to get sick. In order to actually cause sickness, it has to be able to adhere. And so these are going to be things that are going to be such as adhesins. So adhesins are just a generic term for different proteins that are responsible for helping the pathogen actually establish adherence to the host cell receptors. Remember, we talked about having all kinds of different receptors, and that there are receptors that are very specific to different cell types. And so most pathogens are only going after one type of receptor. They're not infecting every single cell in your body. They're actually only going after one, much like we talked about with HIV going after CD4. So in this case, the receptor is going to be specific to the adhesin. The adhesin is going to be looking for that receptor. The majority of the adhesins are going to be located at the tips of the pili. So remember the pili, I tell you, it's kind of like Velcro. That pili that's surrounding the bacteria acts as the Velcro or the adhesin to essentially attach to whatever the cell surface is it's looking for. Colonization. So once it's actually adhered, the next step is it has to grow in the high numbers. So colonization is going to be multiplying and establishing an infection. Then one bacteria is typically, actually, I can't think of a single case where one bacteria is enough. So once it gets inside, maybe only one or two establish adherence, but then they have to start multiplying and get up in the high numbers. That's colonization. Lots of different things that happen there. The first one there, and probably the more important one, is the growth of biofilms. So we talked about biofilms, I think, briefly before. Biofilms are the idea that once a bacteria establishes adherence, so let's say along the intestinal tract, it's established adherence, bacteria sit down, and what they'll do is they'll start producing almost like a mucus type substance, and it will build a barrier or shield around the entire colony. And that's what we call a biofilm, and that actually causes a whole nother level of problems for clearance. One, very difficult to get antibiotics into the biofilm. And two, if it actually breaks off, let's say this is actually inside a catheter, instead of it being the intestines, if it was inside a catheter and it forms a biofilm, these big chunks of bacteria can potentially break off and then cause clotting or cause some sort of other blockage down the way. Yeah.
A biofilm like that in like a catheter, it would take a while to develop though. It would have to be like contaminated prior to, not like if you open the catheter and touch it and then use it. It's like
It's going to be one that's unfortunately the different types of health clinics. Sometimes you don't have the right insurance, you don't have the right way to pay. Sometimes catheters get stuck for several days, and that's when we start seeing biofilm problems. Luckily, there's actually knowledge of that now, and from my understanding, it's not as big of a problem as it used to be. But for a while there, if the patient had a way it means to pay, well, then they get catheter.
like
And
if you have a catheter in the hospital.
Yeah,
it
hospitals,
stays, there's like a certain amount of time that you have to
remember, the
change it.
bacteria, replicate every 20 to 30 minutes, so it could be as much as only a few hours. That it could potentially start forming these biofilms, and then it's only a matter of time if it breaks off or not. That's just that's a fear. That's not a guarantee.
I guess my asking, what's the difference between like a toxin and like a virus or an infectious disease?
So a toxin is only going to be produced by a bacteria or some eukaryotics. Viruses never produce toxins. Viruses are always going to get inside your cell, and they're usually going to be just destroying your cells from the inside out. And that's how they're ultimately causing damage. Toxins are going to be some sort of protein that's being produced by bacteria, and some eukaryotes, some funguses produce toxins also. And that actually causes damage. We'll see them towards the end of this. I think the last few slides actually go into toxins quite a bit. All right, so an example here, this is actually one of the very first projects I worked on as a grad student at Tulane, was looking at these type 3 secretion systems. And so it actually is in your book. So this actually goes to Shane's question, is that delivering effector proteins to host cell, how do the toxins even get there? Well, what's kind of interesting is that some bacteria are actually able to produce these needle-like syringes. And these are what we call a type 3 secretion system. This is actually a picture of one. Almost marries quite well to the actual cartoon. You can tell it's a gram-negative, right? Hopefully you're just, this thing's hopefully you're jumping out at you now. You see the double membrane, that has to be a gram-negative. And you can see that there's multiple parts to it. There's actually 4 or 5 different proteins that all get together. It's constructed. And then basically the syringe will go and then stick the host cell. Just like you would with a syringe, you go stick that host cell, but at a much microscopic level. And now it's actually delivering effector proteins. Sometimes it's toxins, sometimes the protein that goes and changes the shape of the cell. Sometimes it's a protein that goes in and causes the cell to then want to phagocytize the bacteria. There's all kinds of, we could go through, an entire semester of different effector proteins out there. So it's not necessarily one size fits all with this one. Yeah.
And you said, like, at that point, if they're getting into the cell, like, if that's the membrane ruffling, right?
Yeah, membrane ruffling. Actually, that would be how salmonella causes type 3. It has type 3 secretion systems and causes the
And
membrane
then
ruffling.
it encloses the bacteria.
Mm-hmm. So penetrating the skin, this is actually pretty difficult to do. This is going to be the idea that typically they have to go in through a cut or a wound. As you can see there, Staphylococcus aureus, one of my favorite bacteria, actually causes cuts, has to get inside through a cut or some sort of wound. It can't just burrow its way through the skin. Things like Yersinia pestis, the cause of the Black Plague of 1300s in Europe, and we still actually have it to this day in parts of America. It has to actually get through the skin through a flea bite. So the flea bites and then actually delivers the bacteria into our bloodstream. And so again, both of those bacteria have no capability of getting past the skin on their own, but they have different techniques to allow themselves to be delivered on the other side. Penetrating the mucous membranes. This is a major entry point for most pathogens. Usually it's going to be some sort of respiratory, digestive. Remember, anything that you bring inside your system, inside your body, the very first place it's going to touch is the mucous membranes. And so a lot of these pathogens have to actually get through the mucous membranes in order to actually establish disease. So directed uptake by cells, this would be what we were just referring to, where the salmonella can inject those proteins, and that's what we're seeing here is this membrane ruffling, where it's actually causing the effective protein to be delivered by that type 3 secretion system is causing the membranes of those cells to change their shape and allowing very easy entry then for the salmonella to get to the other side of the mucous membrane. And so big key part for most pathogens getting penetrating that mucous membrane. So then the next step is one that we referred to a little bit in chapter 14 when we were talking about the mucus-associated lymphatic tissue or the skin-associated lymphatic tissue, is that there's these antigen sampling, right? You know, where they're basically just tasting what's going through the intestines. In this case, remember I said, well, some pathogens actually take advantage of that. Well, that would be this here, exploiting the antigen sampling process. We're just out there testing, checking to see what's going through the intestine, make sure there's nothing dangerous there. Well, sometimes pathogen actually wants that to happen. It wants to be brought to the other side because it's basically a free ride to the other side of the intestines. It gets right through the mucous membrane. And then once it gets to the other side, it has the ability to survive phagocytosis. And we'll cover a lot of these different techniques when we get into the individual diseases. So don't necessarily focus on, again, like I started the conversation out today, don't focus on all the different examples. You wear yourself out. We're going to cover all those examples as we move forward in the different chapters. Focus on the big picture ideas here, exploiting the antigen processing system, penetrating the mucous membranes, getting across the skin. You know, all the different examples will be wearing it out for sure. All right, hiding within the host cell, again, examples of ways that they're able to get into the system, and then once they get inside, mainly through phagocytosis, they can survive. And they're able to actually multiply within the phagocyte. And then once they're at the critical mass, they bust through that phagocyte, and now it's very problematic for the immune system to catch up. While they're hidden inside that phagocyte, the immune system has no ability to see them. And so they're able to build up numbers, and then once they break out, now they actually got an advantage. They're ahead of the game. The immune system now has to catch up to be able to figure out what's going on.
So do they multiply inside the phagocyte?
What?
They multiply inside?
Yeah.
Just
So like things like
exploded?
but that's how viruses do it too. They basically just blow up your cell once they get to a critical mass. And then once you release tens of thousands, if not hundreds of thousands of virus and bacteria, then the immune system's like overwhelmed, right? It's like, oh my goodness, what's going on? It becomes a process for it to actually catch up.
So when it crosses the other side, if they just let it go and can't kill it.
Yeah, so like things like
It just keeps multiplying.
So an example would be chlamydia. Chlamydia actually gets inside the phagocyte, and typically what happens, right, is phagosome and the lysosome will fuse, and then that causes that phagolysosome to, basically chew up whatever was phagocytized. In the case of chlamydia, it actually blocks the lysosome from binding to the phagosome, and it's able to just happily survive inside that phagosome, multiplying up to high numbers, and then it blows up the phagocyte and gets out and moves on to the next cells. Avoiding killing by complement system. So again, step by step, process, right? It's already got through the innate system. The next backup system before adaptive comes on is the complement. Well, if it's able to avoid killing by the complement, then we have to wait for adaptive to come around. Each minute, each hour that bacteria or that virus or whatever that pathogen is has a chance to live a little bit longer is a disadvantage for us or whoever's actually the patient that's actually infected. So each of these little things is just another trick, another little part of its strategy to actually survive and ultimately win. Winning unfortunately means typically the patient passes away. So here, Neisseria gonorrhoeae, the same one that causes gonorrhea, hijacks the host system, binds the complement proteins, and avoids them from actually being able to attack. So in this case, the idea would be that gonorrhea is able to actually take, remember, what is complement? Basically just all those different antibodies, right? They actually start binding to it, opsonizing. Well, I'll just call it NG for Neisseria gonorrhoeae, it actually grabs the FC portion and turns the antibodies upside down. Now, the complement can't recognize them, phagocytes can't recognize it. That's actually a bacteria that's been opsonized because it's upside down. Remember, the FC portion is what the rest of the immune system recognizes. So in this case, avoiding killing by complement, that's one of the examples of how they're able to do that.
So basically, they're trying to avoid opsonization.
Yeah, I mean, they're just trying to avoid being killed.
Yeah.
So there's different examples of it. Yeah.
What's the connection with the serum resistant bacteria? How does that help?
So serum resistant, serum is just going to be the antibodies. And so in this case, they're serum resistant. They might have capsules on them that hide from the antibodies. It could be that they grab the FC portion. Again, I don't want you to get into all the examples just yet because we'll be seeing them as we go step by step through all the different chapters. These are just examples of ways that they're able to do it. Avoiding destruction, we just kind of covered this a minute ago. Avoiding destruction once they get inside. Typically, they're going to block that lysosome from being able to bind to the phagosome. And remember, the lysosome is where all the nasty chemicals are. The phagosome is just what the pathogen is inside once it's been phagocytized. There's really nothing dangerous inside the phagosome itself. It's when that phagosome and lysosome fuse that there becomes a problem. So a lot of times they'll actually block the capability of that happening. Avoiding destruction by the phagocytes continuing. Lots of different examples here of different things that bacteria may have. So capsules, as I just pointed out, the capsule, remember, goes around the outside of the cell wall. So remember, you got your cell membrane, then you got your cell wall, whether you're gram-negative or gram-positive, and then sometimes you'll have a capsule that goes around all of that. That capsule kind of acts like camouflage. Because remember, what is it that the toll-like receptors and all the other different types of receptors found in the innate system, they're looking for those pathogen-associated molecular patterns, right? They're looking for LPS, which would be gram-negatives. They're looking for peptidoglycan, which would be the first thing that a gram-positive would have. Well, if you cover that with a capsule, then you're actually going to avoid a lot of the immediate innate responses that we have already set up. And so having those capsules sometimes can prevent the complement from recognizing and starting the cascade. M proteins, these are going to be ones we'll cover more when we get into the wound and skin infections. M proteins are just ones that bind to the C3b once it actually has already started a cascade. Remember, C3b is the very first step of the cascade once C3 splits. Well, if the bacteria can grab C3b and inactivate it, well, you basically just shut it off, and so the rest of the cascade doesn't follow suit. FC receptors, that's what I was talking about here with Neisseria. Also, a couple others there, Staphylococcus and Streptococcus, can also bind the FC portions of the antibodies and basically flip them on their opposite side. And now the rest of the system can't recognize it. Sure, complement may be throwing out tons of antibodies, but if the phagocytes can't recognize it, then it's basically shut down there too. So each of those options are all just as effective. Notice Streptococcus pyogenes actually has two of them. Has the capability of inactivating C3b and also has the capability of grabbing the FC portion. And so that's when virulence factors become important. They're always important, but when you have multiples on a single pathogen, then it becomes very important for us to truly understand what somebody's infected with so we know what weapons that particular pathogen has available to it and then how to optimally combat them. Some more here. Escape from the phagosome. Listeria monocytogenes, which is the one that Bluebell got in trouble for a couple years ago. It shut down the entire Bluebell factory for a minute. Listeria is a very interesting bacteria. It's one of the only ones that infects humans that actually prefers living in the cold or can survive in cold. So that's why it was important for dairy products as well as, ice cream's a dairy product, but yeah, pretty much any sort of refrigerated product can potentially have Listeria, but we usually see it in dairy as well as meats sometimes. And usually it doesn't cause a major problem, except for it is problematic for pregnant women that get infected. So that's where we usually see the biggest problem. But also elderly, anybody that might be immunocompromised. Listeria, once it gets phagocytized, it is going to be able to escape before the lysosome fuses. Right? So every step of the way, there's an example of a pathogen that's able to avoid one part or the other, is hopefully what you're seeing here. Is that when we learned all this in chapter 14, we've seen that there's a process. Well, every step of that process, there's an example here of something that's able to avoid that part of the process. So here, Listeria as well as Shigella are able to escape from the phagosome. Basically, they just break out and get away before the lysosome ever has a chance to bind. Next one down, what's preventing the binding of the phagosome lysosome? So in this case, it's staying inside the phagosome, but it's not able to actually allow the lysosome to bind. And then very few, interestingly, but there is one there, Coxiella burnetii, that is able to survive within all that thrashing, all those different enzymes, all those different toxins that are found inside the lysosome. This particular bacteria really doesn't seem to be bothered by it. It has the capability to survive all that also. So even if the phagosome lysosome fuses, it's able to survive. And so again, luckily that's not one that we see very often. It's kind of more of an exotic disease. Exotic meaning that it's pretty rare, but it is an example one that's able to survive those phagolysosomes. IgA proteases, some of them are going to come equipped with a protein that actually splits IgA. Remember, IgA is a dimer, right? So it's actually two antibodies bound together, and that's how it operates at its most efficient. And so if you actually have a protease, remember ASE is an enzyme, right? So it's an enzyme that chews proteins. Antibodies are proteins. And so in this case, an IgA protease is an enzyme that goes and just cleaves the IgA and splits it in two. If it's able to do that, now IgA is not able to do its job. And so in this case, Neisseria gonorrhoeae and others are able to actually just cleave IgA, and now it's able to survive that initial mucous membrane attack that IgA is typically part of. Antigenetic variation is also pretty interesting, where they're able to change their colors. So it's sort of like somebody goes and robs a store and goes outside and changes their outfit, so that way they look different. And hopefully, the eyewitness accounts are like, oh well, he was wearing or she was wearing this. Go out and change your clothes immediately. You look different. In this case, some bacteria and even some viruses are able to change their antigens on their surface. Remember, the very first thing that the immune system is going to see are going to be what's on the surface of that particular pathogen. And so if I could change what my surface antigens look like, the immune system is going to have a much more difficult time immediately recognizing that particular pathogen and knowing that there's a problem, especially if it's already been chasing one variation of it, and mid-pathogen, mid-disease, it changes its antigens, and now the immune system basically has to start all over again, developing an adaptive response, because that response it was making isn't what that pathogen looks like anymore. And so there's different examples of that. We have gonorrhea up there yet again, but another example from a viral standpoint would be things like influenza. We call them H1N1 or H5N1. Those are the two antigens that are found on the surface: hemagglutinin for H, neuraminidase for N. And so, every year, why do we have to get a new flu shot? Because it looks different. The antigens on the surface are different. And now whatever that particular flu shot you had the year before is going to be different than the flu we have this year. And so most people are like, why do I have to get a flu shot every year? That's why, because it's actually changing what it looks like every year. And that's just part of its natural evolution. Some years it's mild, it doesn't change that much. So you could actually, if you had a flu shot and you forget to get it one year, you'll say, oh, I didn't get my flu shot this year. I didn't get sick. Well, congratulations. You won the evolution battle that day.
So do you really believe in the flu shot?
Absolutely.
I've like never gotten the flu shot.
Really? Well, you're young, so your immune system's been fighting it off just
But
fine.
do you completely believe in the flu shot? Because I've heard like, people say weird things, but people say weird things about it. So.
Well, when we get to the flu, we'll tell you the whole story of how that came about, the hoax of how vaccines were shown to be causing autism and whatnot.
Well, no,
Yeah.
I know that, like I never knew you. That's a different subject. But I was talking about the flu shot.
It's all the bullet people relevance, yeah, exactly. But yes,
It's
yes.
just like a vaccine, like getting when you're little, like it would just be like that.
Yeah, so, but the thing is, why do we have to get it every year? In a genetic variation. Things like, so why do we not have to get measles, mumps, and rubella, but maybe once or twice? You get your 6 month, and I think your 1 year, and then there's a third that the CDC recommends as you're an adult, you should get an adult booster of it. Why? Measles, mumps, and rubella, those only have one pair of clothes they wear, right? So there's no antigenic variation. So that particular vaccine will last long periods of time, presumably. Again, we're still in the infancy of understanding how long memory cells last. And so that's why they just recommended in the last few years to get that booster for MMR. Also boosters for DPT and others. But ultimately, the antigenic variation is why things like flu, you have to get the shot every year, is that it changes what it looks like. All right, mimicking host molecules. This is one of my favorites in terms of the way that they're able to avoid host defenses, is that they just go and start looking like you. So they'll actually change what their shape is on the outside. They'll basically cover themselves in some of your cell. So imagine that they break out, if they've been phagocytized, it's just one example. They break out of that phagocyte. Well, now they got pieces of that phagocyte around the outside of them. The immune system's going to now recognize it as self for a minute, at least, while again, it's all strategy, right? That may last for a few hours, may last for a couple of days, and then finally the immune system's going to figure it out. But at the end of the day, every minute, like I said, every hour that pathogen is able to survive is another problem for the immune system to ultimately clear. All right, so direct and indirect effects. So those are all different examples. Those 3 slides are different examples of how it's able to survive phagocytosis. Again, just every single one is an example. There's no such thing as a bacteria or a pathogen that has all of them. It's just different examples that we've been able to identify. Here now we're switching gears. We're talking about damage to the host. You can have direct effects, which are typically going to be toxins. The toxins will do a direct effect, whatever that toxin binds to. Typically causes damage. Sometimes the toxin can cause permanent damage. Sometimes it's temporary, depending on which toxin we're talking about. But those are direct effects. That toxin is what is actually causing the damage. Indirect effects are going to be things like evading the immune system, manipulation of the immune response, like we just went through just a minute ago with the last 3 slides. Those are all indirect. They're not necessarily causing damage, but it's all a means to an end of ultimately causing damage at the end of the day. Toxins are direct. They're causing damage immediately. Those other ones are causing damage, through the process of pathogenesis. So damage may help pathogen exit and spread. So one of the things we'll cover when we get to digestive diseases, we'll cover it right now, but then we'll see it in more glory, as we get into digestive diseases. Vibrio cholera is known as a 2-bucket disease, mainly because you're throwing up and having diarrhea at the same exact time. Why would that be a strategy? Well, because it helps spread, right? Especially in places in third world countries where they may not have the best sewer treatment facilities. You may have a lot of family members living in close proximity in the same home. If you have somebody that is having a cholera infection and they're having, imagine that you're probably not catching everything, right? There's going to be a lot of spillage, there's going to be a lot of problems. And in this case, it's going to allow for that bacteria, Vibrio cholera, to be able to spread faster from person to person. Somebody that comes in contact with that vomit or that diarrhea may potentially contract it themselves. So that's the biggest fear we have for a cholera outbreak, is typically the spread. The spread is very quick sometimes. As you can see there, 20 liters per day. Somebody that has a very serious infection. So let's think about that. It's about 4 gallons. About 4 liters is 1 gallon. 5 gallons of fluid. How much does a gallon weigh? About 10 pounds roughly. So you imagine that somebody that has a full-blown cholera infection is losing about 50 pounds of water weight a day. You're not going to last very long, right? You're going to be dead pretty quick. So the big key to people that have cholera infections is just rehydration. There's really not much more we can do for them besides rehydrate. It's pretty nasty to be involved in. Bordetella pertussis, the cause of whooping cough. Why does it have the name whooping cough? The sound. So the problem is somebody that's coughing, so especially infants, right? It's a big problem for infants that get pertussis. And so when they're coughing so hard, they can't catch their breath. And when they finally get a chance to catch their breath, it's, well, that's where it gets its name whooping cough. It's terrifying to see a child going through whooping cough. For an adult, it's much more mild, but for an infant, it can be fatal pretty quickly. Well, why is that coughing fit so important? Well, that's part of the evolution of that bacteria. Coughing so hard and violently is throwing that bacteria as far as possible to hopefully be picked up by another person, ultimately, and then to spread. So damage may help pathogen exit and spread. Exit the person that's infected and then spread to the next individual. Exotoxins. So when we're talking about toxins, when I talk about all these different types of toxins that we're going to see as we go forward, we're talking about exotoxins. Exo means that it exits, right? So it exits the bacteria or exits the fungus that's producing it. So we usually use the word secreted. So secreted is basically you're making that type 3 secretion system and secreting it through that, or it has some other sort of a secretion system that allows it to move the toxin from the bacteria out into the tissue. And so this could be, like we said, with Clostridium botulinum. It could be that it's producing a toxin into food, and you may not actually ingest the bacteria, but if you ingest a toxin, it's enough to cause disease symptoms. Most toxins are going to be heat sensitive. So again, that's one of the biggest reasons why the Retor systems that we talked about in chapter 5 that we use for canning, they involve heating up. That's why we also talk about pasteurization and other ways of heating things. A lot of times these toxins are pretty weak in terms of being able to be destroyed pretty easily. And so if we're thinking about canning food or some sort of food preservation, a lot of times we heat it up or cook it first, right? We typically don't store raw food in cans. It's going to be already processed and cooked to hopefully get rid of those toxins if they were there in the beginning. Can act locally or systemically. So a toxin we're talking about can be localized. It can just start chewing up tissue right around the site of the infection. This is going to help the bacteria continue burrowing deeper and deeper into the sterile tissue. That would be localized. Systemic is going to be something where you have an infection maybe in your toe, but it's producing a toxin. That toxin is getting into your blood and then going systemic, and then you have serious problems, usually with shock and whatever maybe the toxins byproducts from there. The proteins, so the immune can generate antibodies. So all toxins are proteins. So remember, proteins make the strongest antigens. When we're talking about the antigen response, proteins are always the most important. And so exotoxins being protein makes the best, makes it really easy for us to actually produce antibodies for it. But the problem is we got to think about how long does it take that adaptive system to come online? 10 to 14 days. You may not have 10 to 14 days with a toxin. And so then that becomes the win for the toxin anyways. Our immune system doesn't have the time to catch up to it. So that's why things like vaccines are important for that too. So diphtheria, part of the DPT vaccine, diphtheria produces a toxin. So it's better to actually already have the antibodies ready to go against that bacteria, so that way our immune system can shut it down immediately. Because if you actually establish an infection and it starts producing a toxin, then you're going to be already behind. It may be really difficult to clear that particular infection again.
I don't know a bunch about it. I just have a gut feeling that it's all bull crap. But how there's all these things going around rid your body of toxins. Wouldn't, if your body was full of toxins, wouldn't you be dying? So you don't really have
So those are that's a lay version of toxins. So they're referring to, here we're referring to toxins being produced by bacteria. When people are talking about detoxifying their body and whatnot, they're trying to get rid of all the different types of toxic chemicals that may be part of the preservatives that are in the food,
Oh, okay.
things like that. So, eating healthy, eating clean, blah, blah, blah. Is there anything wrong with eating clean? But is there anything wrong with preservatives? Yeah, it depends. There's some interesting stories out there from preservatives where, I think I may have told you this and not, I'll tell you again, before the FDA was able to regulate a lot of these preservatives, they were putting tons of them in products. And there was one person that kept saying, these are bad. We can't, and nobody wanted to listen to him. So he decided to start a dinner, a weekly dinner with all his friends that were against the idea that preservatives are bad. And he just loaded their food up with these preservatives, unbeknownst to them, and would serve it to them at dinner, and they would all get sick. And then he would go, okay, have you all had enough? Now are you going to listen to me that these preservatives are bad? So the question is, my master's is in toxicology. The golden rule of toxicology is anything is dangerous. It just depends on how much of it you have. So even water can be toxic. Yeah. And so how much of it did you eat? Are you the type that eats nothing but preservative foods every single day? Then you might have a problem. Somebody eats something every now and then. I don't think the toxins that, if they are truly toxic, I don't think they're building up the levels that are dangerous for you. So a lot of that's a marketing scheme.
Different types of toxins.
Absolutely.
Because when I hear toxins, I just think of your body's full, you're dying.
Mhm.
And
Well, that would be an exotoxin for sure. But people have mercury toxicity, arsenic as a toxin. Those are different. Those are heavy metal toxicity. Here we're talking about toxins that are produced strictly by the bacteria, the fungus.
And that bacteria can be gram-negative and gram-positive. They all make it, right?
What?
I think it's
I'll bite my tongue on that question, but I'm pretty sure the majority of them are gram-negative.
It says a number of gram-positive and gram-negative.
Okay, then then it doesn't matter. I was just trying, the immediate ones that were popping over my head were all gram-negative.
I was unsure about
Yeah, then they both can, absolutely. All right. Okay, where are we at? So many fatal before immune system mounts its response. That was what I was just saying there a second ago, right? So ultimately, once you get the infection, then it's hard for the immune system to actually catch up if it starts producing the toxin. So we have the vaccines as well as antitoxins. So if somebody comes in and they have an established infection, a lot of times we have the antitoxins available, and that's the first thing we need to do. We're not worried about giving an antibiotic to clear the infection. We're worried about getting that toxin out and at least trying to shut the toxin down first, and then we can deal with the actual cause of that toxin. And so there are different types. We have neurotoxins. These are ones that are going to damage the nervous system, so they shut down nervous system. So it could be things like the botulinum. It actually causes flaccidity of your muscles. So people that got wrinkles and whatnot, they go get Botox injections. What's it actually doing? That toxin's going in and attaching and actually causing flaccidity, causing the muscles just relax. And if that happens throughout your entire body, well, you're going to have respiratory distress. You're going to have potentially heart arrest, right? Because the heart's a big muscle, so essentially the toxin can get in there. It hits the neurons, and then it can shut down the muscle tissue. Enterotoxins, these are kind of causing intestinal disturbances. So this is where a lot of diarrhea, especially when we talk about some of the more nastier ones like cholera, causing much more serious diarrhea called dysentery. So the difference between diarrhea and dysentery, blood in the stool. So if you have blood in the stool, it's dysentery. That's usually going to be toxin related. If it's actually destroying the intestine so much that blood's coming across and you're finding blood in the stool, then that's going to be probably toxin related. Cytotoxins, we just call those generic, right? Cyto is just cell. So if it's just causing generic damage and it's not specific to a type of cell, then it's cytotoxin. But just like the bacteria, just like the viruses, where they have those adhesins that are cell-specific. A lot of times the toxins are cell-specific too. So that's where we get things like neurotoxins are specific to neurons, enterotoxins being specific to the intestinal cells. Yeah.
Or even definition of the antitoxin that says suspension of the
So suspension is because it would be ready to inject. But the antitoxin itself is going to be antibodies. So we go back and you look at the antibody slide from chapter 15, where we had all the different ways that antibodies can work. There's like 7 of them. One of them was binding the toxins, and that was basically shutting them down. So at the end of the day, antitoxins are antibodies for the most part. And so they're going to bind. And so it says suspension of neutralizing antibodies. The neutralizing antibody is the antitoxin suspended in some sort of solution that allows it to be injected. All right, so these next 2 slides are going to be ones that you can go to just to kind of refer to all the different types of toxins out there, exotoxins, and the bacteria that produce them. Again, nothing that you're going to need to know for this test coming up on Tuesday, because we'll be covering all of these, every single one of those, I believe. Yep. Every single one of those we're going to cover at some length in different chapters. So don't necessarily memorize them right now, but if you want to get ahead of the game, you can kind of start getting an idea or a feel of how a lot of these different bacteria can cause disease. Here, some more. So we go all the way from Clostridium perfringens all the way down to Staphylococcus aureus. So we'll see all those coming up chapters. All right. So back to his question of what is a toxin in the first place. Well, most toxins are what we call AB toxins. It means they have 2 parts to them, an A and a B subunit. The A subunit is actually the toxin. So if I was to draw it out here, I just draw it as 2 parts. The A is going to be the actual toxin. B is the subunit that's binding to the receptor. So what we're seeing up there, right? So it's just really just 2 parts, and that's what makes them cell specific. The B part, the B subunit, is what's making it cell specific. A part is actually the toxin. So what happens, as you see here in this little cartoon, is you got the AB toxin here. The B is binding to the receptor on the cell surface, whatever that receptor is that that B subunit recognizes, causes phagocytosis, and basically you just brought the toxin right into the cell. And now that toxin is able to do its job, kill the cell typically, cause some sort of ruffling if it's the membrane ruffling toxin. But almost all exotoxins are AB. Again, something evolutionarily there is relevant, where they're actually, even though we have lots of different bacteria that are not related per se, they all produce an AB toxin that looks very similar. One of the things that it says there towards the end is that sometimes that B subunit is important. What I've been saying all semester is that a lot of times we don't actually develop anything novel when we're coming up with new therapies and new pharmaceuticals. What we're doing is taking advantage of what nature's already given us. So what we can do is that the A and the B are actually, they have a DNA portion to them. They have a gene, right? It's actually responsible for making it. It's a protein. So that means that there's a DNA component to it, a gene somewhere that codes for that protein. And so what we've been able to do in the laboratories and in pharmaceutical companies is we actually just use the B portion, get rid of the toxin, and we add whatever drug we want to put on there. Now we got a beautiful delivery system. Imagine that. We actually have a delivery system that we can utilize that is very cell-specific. And so now we can take that B subunit, we don't touch it because it's good. It actually binds to the receptor. Instead of it delivering a toxin, we now have it delivering a beneficial drug of some sort. So one of the ones I was working on at the Primate Center was one called ONTAC, and ONTAC was a new drug that basically was designed to use the AB subunits, use the B subunit to bind to T cells, and then we delivered a drug, as a potential HIV, as part of the HIV cocktail. I don't know if it ever got approved or not. We were only doing a couple little projects with it, but it was an example of an AB toxin. They're definitely out there, in terms of using the B subunit for good, getting rid of the toxin, replacing it with something better, or some sort of therapy. So different examples of toxins, we've seen them already. Membrane damaging toxin, going in and damaging the membrane of the cell. When you do that, you're going to cause the cell to lyse typically. So remember, when you lyse a cell, you basically split it, kill it. And so here's cytotoxins that disrupt the plasma membrane. If you disrupt the plasma membrane too much on our cells, remember our cells don't have a cell wall around them. All they got is the phospholipid bilayer. You start damaging that phospholipid bilayer too much, you're going to end up having the cell basically just die because everything spills out. Hemolysins, these are very specific toxins that only bind to blood, right? Hemo. So hemo's blood, binds to it, destroys, lyses the red blood cells, starts causing some serious problems for individuals that are infected with. That's going to be things like Streptococcus has a hemolysin toxin. Some are going to insert into the membranes and form pores. When that happens, it's going to cause everything to come to the outside too, kill the cell. Phospholipases, again, into the ASE, it's an enzyme. Phospholipid would be the actual portion that's making the membrane, right? So a phospholipase is an enzyme that chews up phospholipids. So in this case, that would also basically mean the same thing. You're damaging the membrane. That's why all 4 of those bullet points are tabbed over for membrane damaging toxins. They're just different examples of toxins that we know exist, but different methods for how they actually cause the damage. One of the more interesting ones is the superantigens. These are going to bind to the MHC class 2 and the T cell receptor at the same time. Confuses the T cell into believing that it's actually got the right antigen. Remember the T helper cells. We're talking specifically about the T helper cells here. They actually, need the MHC class 2 binding to the antigen presenting cell, and then that T cell receptor also has to give the next positive feedback. Well, sometimes, as you can see here in this picture, you may have the wrong peptide in you. Here's when it's right. Here's when everything's going right. Here's when it goes wrong. You got the wrong peptide in there. It's not what we're supposed to be going after. In that case, antigen, we'll call it an antigen. It's got the wrong antigen in there. It shouldn't bind to it. The T cell should tell it no. But instead, this toxin comes about. It's called a superantigen toxin. Binds to that receptor and goes ahead and gives a positive signal. It essentially overrides that process that should have told the T cell never mind, or told the antigen presenting cell never mind. But instead, it pushes it forward and tells it, yep, that's what you wanted. And what that leads to then is all kinds of chaos happening. It basically causes the immune system to get overexcited because this could be happening over and over and over again. The T cell, it's not going to just happen once. It could be multiple different T cells that are all just testing antigen presenting cells that have things in them that they're not supposed to be responding to. And if that superantigen is being produced, in this case by Streptococcus aureus, it's going to bind and cause major inflammation, because what's happening during the immune response? Lots and lots of inflammation. Too much inflammation is always a bad thing. Leads to things like toxic shock syndrome, which we'll talk about later. But this is going to be an example yet again of how a different type of exotoxin can work. Some other examples there, again, don't get into the weeds of them because, as I keep repeating, we're going to see them more and more as we move forward. Some of them are going to damage proteins. They're not AB, so just to keep in mind that not all toxins are going to be AB toxins, that there are some other examples out there. For instance, exfoliatin. Notice Staph aureus keeps showing up. That's why he's my favorite. He has tons and tons of different weapons at his disposal, different virulence factors. So exfoliatin is one that allows it to basically chew through the skin. I already said it's hard to get through the skin. Most of the time it has to be brought to the other side to get it through some sort of insect bite or some sort of wound. Some variations of Staph aureus actually produce a toxin called exfoliatin that allows it to chew right through the skin. And this is something that's typically problematic for newborns. And so we see the term there, scalded skin syndrome. We traditionally don't see that in older patients, but in children that have a staph infection, it actually looks at the entire body, looks like they've been burnt. And that's because of all the inflammation from the exfoliatin, basically causing the skin to fall off, makes them look like they have like whole body burns on them. So pretty sad little thing. Various hydrolytic enzymes. Hydrolytic enzymes are going to basically chew up something. So in this case, it could be proteases, again, generic enzymes that chew up proteins, lipases, enzymes that chew up lipids, collagenases, enzymes that chew up collagen. Remember, collagen is your basement membrane holding everything all together. So if it's chewing up collagen, it allows it to be able to push through to the deeper areas of the body. All right. Those are all exotoxins. That means they're secreted by the pathogen. Luckily, there's only one endotoxin. Endo means it doesn't get secreted. It just happens to be part of the bacteria. Problem is, the only endotoxin is LPS. LPS is on every single gram-negative bacteria. So, our body recognizes LPS as a toxin, causes damage. Remember, you have to go back and look at chapter 3 again. They look at what LPS looks like. But here we talk about the different portions of it. Lipid A, which is the tip, or it's the middle part actually, triggers an inflammatory response. It causes inflammation. That's what causes it to be a toxin. It's causing inflammation. If that gram-negative bacterium gets into your bloodstream and it's causing inflammation, it's not localized anymore. It's not in one little spot. Say like you get an infection on your leg, it's localized. If that bacteria is able to get into the bloodstream and start circulating, the immune system's chasing it. And it's constantly producing inflammatory response all over the body, which is what we see as being a systemic inflammation. Systemic inflammation is going to cause death pretty quickly in somebody that's not being treated. My mentor actually went through it last year. He got a staph infection that went systemic. Systemic infections actually cause septic shock. What's happening there? What happens with the inflammatory response? Putting it all together from chapter 14 and 15. Remember what happens is that inflammatory response causes dilation of the blood vessels and allows the immune system cells to get to the other side. That's great in a localized infection. If you're causing dialysis or dilation of your entire circulatory system, that's bad, right? All of a sudden, you're going to have a massive blood pressure drop. You're going to end up going into shock, which is the next step of a major blood pressure drop, and that's going to lead to death if it's not contained or not treated well. So this is why LPS is a major problem. LPS, again, it's one of those pathogen molecular patterns, right? It's one of the pathogen-associated molecular patterns, the PAMPs. So our immune system's already ready and ready to go if it sees LPS. In this case, if it gets into your bloodstream, that's where you get the systemic inflammation become very problematic. Also, if it's cell lysis, so let's say we destroy that bacteria. Well, if we destroy it, there's still pieces and parts of LPS as part of the broken up bacteria. That can also lead to inflammation, as well as activating the innate and adaptive responses, like we just said, because their PAMPs can activate it. All of it is very difficult to control once that snowball starts with the immune system response. Heat stable, unlike exotoxins, so yin and yang, right? Exotoxins are very heat sensitive, so they're easy to destroy. LPS, not so much. Autoclaving, it doesn't necessarily destroy LPS. So when I was working in a gram-negative bacteria lab, we really had to go through extra layers of cleaning to clean the glassware. We actually eventually just went to completely disposable. Not because we're worried about us, but it screwed up our experiments. If I was trying to do an experiment where we weren't using a gram-negative and grew it in the same flask that we just grew gram-negatives in the day before, if there's some LPS left in there, it would give false results to our experiments. All of a sudden, our experiments would look like they were getting systemic infections instead of what we were suspecting them to get. And so that's where it's talking about autoclaving does not destroy it. That's typically something that's important for laboratories. And again, one of the hardest things for my biotech students to understand is that they start freaking out. They're like, well, why are we always using all this disposable? They feel bad throwing away all these plastics and whatnot. And yeah, I get it. But the alternative is much worse, where you're actually getting bad data and things aren't working well. So a lot of times it's more economically in the long run to actually use disposable over recyclable things. So peptoglycans and other components also trigger inflammation, right? the LPS has a very good knack for really inciting it. Therefore, it is the only endotoxin. All right, so comparison of exo and endos. Exotoxins, as we just pointed out, come from both gram-positive and gram-negatives. The proteins, always, they're potent. They can do their job very well, but they can usually be destroyed pretty quickly by heat. And so that's going to be important for food production and things like this. Endotoxins only from gram-negatives, right? It just stands to reason because gram-positives do not have LPS, and so they can only be gram-negatives. It's almost redundant to say. So in this case, we got some different examples here of exo and endo, bacteria source, location, chemical nature, ability to perform, blah, blah, blah. You can go there and look at those just as comparison purposes. All right. Damaging effects of the immune response. Well, this goes through everything that we've already talked about, where sometimes the immune response is the problem. It's not necessarily the pathogen. Sure, the pathogen's causing the immune system response, so you can challenge me on that. But what I'm trying to point out here is when we actually look at the pathogenesis of a particular disease, sometimes it's actually the immune system's fault for some of the signs and symptoms we get. So damage associated with inflammation. Kind of went through that, right? We go through and actually can cause shock, cause all kinds of other damages, in terms of like it could be, as we'll see here in a minute, it could be things like immune complexes that get formed, cross-reactive antibodies that cause autoimmune diseases. There's a lot of different examples there. But damage associated with inflammation specifically is going to be involving the neutrophils. Remember, they're dumping all their granulocytes. That's going to be all the different enzymes that they have and different toxic-like compounds that cause damage to the pathogen. It's also going to cause damage to the host cells, too, that are surrounding that area. Specifically to the adaptive immune response, you have immune complexes. So remember when we talked about some of the good things that antibodies can do, since they got two arms, they can grab one bacteria and they can grab another one, and that causes all this, basically this big clot of bacteria where they're unable to do their job anymore. They've either been immobilized or been blocked. Well, if you have a lot of that going on in a very tight spot, like say in a joint, it can cause problems. Those complexes actually accumulate and cause, arthritis. And so we have things like, arthritis is one. It's not up there. The one that you're pointing out is a nephritis, which is an example of a kidney. So when those immune complexes get into the kidney, they actually block all the different pores inside the kidney and cause the kidney function to start decreasing because they're blocking up all the different passageways. Cross-reactive antibodies, that's referring to the idea that sometimes those antibodies, especially in a major response, we talked about lupus being one, another example here being staph pyogenes, that actually causes rheumatic fever. And this is going to be an example where those cross-reactive antibodies then may be going after the pathogen, but those antibodies may also be able to cross over and attack self too. Or sometimes a mistake is made and self-reactive antibodies are produced. And that's going to be, a side product of the actual disease. Binding the host cells and invasions. So here viruses can target, change the subject. Mechanism of viral pathogenesis. Everything we covered up to this point was mainly bacteria, but I didn't want you thinking that there was nothing else besides bacteria that can produce toxins. There are a few funguses that can too. But no viruses, right? So here, viruses are just the last three slides here. Binding the host cell and invasion. The biggest thing that a virus does is it gets inside your cells. Once it gets inside the cells, the immune system can't see it. That's where the MHC class 1s and the NK cells come into play. It's ultimately in CD8s, right? Because they're going after the viruses. Avoiding the immune response. Well, if they're hidden inside the cells, it's really difficult for the immune response to have any effect. We also have avoiding the antiviral effects of interferons. So back to chapter 14, interferons are produced by the cells to block viruses' ability to multiply. And so if they're able, some viruses actually have onboard proteins that block interferons. Again, evolution, nature finds a way. Some of them actually are able to block the interferon process. Some of them can block and avoid antibodies, depending on which virus we're actually talking about. and regulating the cell host death. And so in this case, preventing or delaying apoptosis. One of the biggest proteins that's involved in the process of apoptosis, the decide or not decide to go through apoptosis protein, is called p53. That's why it's up there. That's the regulation protein, so it's regulating whether we go through apoptosis or not. Some viruses will actually block p53 from being able to do its job. And so even if it's getting a signal from the NK cell, getting a signal from the CD8 cell to go through apoptosis and destroy itself, some viruses say, nah, we're not doing that, and actually keep that cell alive a little bit longer by blocking the capabilities of the go through apoptosis. Some of them will block MHC class 1's presentation. That's when NK cells come in. Remember that NK cells only respond if the MHC class 1 is missing. Well, that takes time to be able to recognize. And so by hiding the MHC class 1, the virus is able to multiply up and possibly spread before that MHC class 1 is recognized to be missing by an NK cell. And one of the more clever things that some viruses can do is throw up counterfeit MHC class 1s. That way it makes it look like it has an MHC class 1, even though it's a make-believe or a fake that it actually throws up there. Again, none of these are an end-all be-all, but depending on how many of these different virulence factors a virus or a bacteria has on board can cause differences in how serious that disease is, right? If it's, or how fast that, somebody needs to seek medical attention. All right, and then eukaryotic pathogens, again, nothing serious per se when it comes to eukaryotic pathogens. Most eukaryotic pathogens are causing self-limiting or annoying at best diseases, but there are a few that are fatal or can be fatal. So funguses, they're going to be, most are what we call saprophytes. And so saprophytes are ones that cause disease only through opportunity. So some sort of wound, somebody that isn't very well kept can get a lot of skin infections from funguses. Dermatophytes are going to cause superficial infections, hair, skin. So if people have got nail fungal infections in their nail or fungal infections in their hair, cause the hair to fall out or cause yellowing of the nail. Fungus is a normal microbiota, so Candida albicans we talked about before. That's the main yeast that causes yeast infections. So traditionally in females, they get yeast infections. It's almost always Candida albicans. The other two there, Aspergillus fumigatus. This is black mold or a variation of black mold. So we had this major problem after Katrina. Lots of black mold throughout the city. People were breathing it, had a lot of allergy problems, a lot of, some people were actually having more serious problems. Cryptococcus neoformans. This is the one that I was talking about that's found in pigeon dung. So living in an urban environment, it's the one that we breathe in quite a bit. But doesn't really cause any problems unless you're immunocompromised, right? If you become immunocompromised, it can cause death. It's like, I think it's the 7th or 8th leading cause of disease in AIDS patients.
All of those can cause death?
What?
All of those can
if you're immunocompromised, for sure. Because the immunocompromised means that they're able to grow uncontrolled, and then that's going to cause all kinds of damage in the body. But for most healthy people, you'll never, I won't say never, because never is always a terrible thing to say as a scientist, but Cryptococcus neoformans infections we don't really see in healthy people. Aspergillus, usually causes mainly at best allergic reaction type symptoms, and Candida albicans at best causes mild but yet irritating yeast infections. But anybody that's truly immunocompromised to the point where they don't have a strong, if at all, immune system, potentially could cause serious problems down the road. So most serious fungal infections are caused by dimorphic funguses. So this is going to be things that change their shape. So we're like thinking about a caterpillar turns into a butterfly, right? But think about it at a fungal level. Some funguses you breathe in as spores or as some sort of mycelia, and then it gets inside your body and actually then causes switches its morphology, and that's where it becomes problematic. There's only a few of them, as I was pointing out earlier, that are actually able to produce toxins, and so we call those myco. Anytime you see the term myco in front of something, Myco just means fungal. And so mycotoxins are going to be toxins that are produced by funguses. And I think that was the last slide for this particular chapter. Oh, no, one more. Protozoas and helminths. Protozoas and helminths. Helminths, remember, these are worms, so these are going to be worms that cause disease. And again, most of them are not going to cause any sort of serious disease outcomes. But they do have some techniques that allow them to survive in you for a while longer. Some people that get worms, they actually carry them for months, if not a year or better, before they recognize that they have a problem. They all have the ability to avoid the immune system, but I'm never asking any questions on eukaryotic pathogenesis on this chapter. So those are up there just for you to have as comparison points, right? Mainly focus on bacteria. I might have one question on viruses, but for the most part, we talked about bacteria mainly in this chapter. Remember also, focus big picture, right? Don't get into all the examples or you're going to wear yourself out. Focus on big picture ideas because that's going to be the type of questions you're going to have. I got 50 questions on the exam, roughly about 16, 17 questions per chapter. I'm not asking minutia, right? Focus on the big picture ideas. Studying in the book, look at the major topics, in terms of the titles as well as the subtitles for each of the different sections. Make sure you understand how all that stuff makes sense. I have office hours from 9 to 11 on Monday. And also 8:30 to 9:30 on Tuesday if you want to talk to me before the test. Otherwise, good luck studying. We'll see you all here on Tuesday. It'll be a paper-based exam, unlike the last one was computer-based. So bring a Scantron. If you don't have one, I'll have one for $1. But go get them in the bookstore. They're like 50 cents over there. Just any. I pay for them. Right? I pay for them. I pay for them. So ultimately, bring a Scantron. If you don't have one, I'll have them available. Hey, I go into debt actually. buying them.